Hello all, today we'll see about nerve physiology. Nervous system is divided into central nervous system and peripheral nervous system. Central nervous system contains the brain and spinal cord, and peripheral nervous system is further classified into autonomic nervous system and somatic nervous system. Autonomic nervous system is further classified into sympathetic, parasympathetic and enteric nervous system. This peripheral nervous system contains the nerves emerging from the central nervous system. Neuron. Neuron is a structural and functional unit of nervous system. Structure of neuron. Neuron contains the cell body, dendrites and axons. The cell body contains nucleus, Nessels granules, neurofibrils, mitochondria and Golgi bodies. The axon may contain myelin sheath. Axon hillock is the junction between the cell body and the axon. This is the location where action potential originates. Cross section of a nerve. It contains nerve bundles called fasciculus. Inside the bundles, nerve fibers are present. There are three layers of covering: epineurium, perineurium and endoneurium. Now, classification of neurons. Based on myelin sheath, they are myelinated and non-myelinated neurons. Myelinated nerve fiber contains myelin sheath, which is a lipoprotein membrane which insulates the nerve fiber. The cells which help in myelin sheath formation are schwann cells and oligodendrocytes. Schwann cells in peripheral nervous system and oligodendrocytes in central nervous system. Myelin sheath also helps in solitary conduction of nerve impulse, which is the faster method of conduction of impulse. Based on function, the neurons are classified into sensory neurons and motor neurons. Sensory neurons carry sensory impulses to the brain, and motor neurons carry motor impulses to the effector organs. Based on the number of poles, the neurons are unipolar, pseudo-unipolar, bipolar and multipolar neuron. In unipolar, a single process extends from the cell body. Example, neurons present in the dorsal root ganglia. And in pseudo-unipolar, a single axon emerging divides into two branches. And in bipolar, there is an axon from the cell body and a dendrite from the cell body. Example is the bipolar cells present in the retina. A multipolar neuron contains many dendrites from the cell body and a single axon from the cell body. Example, many of the neurons are multipolar neurons in the brain. And the multipolar neuron is further divided into Golgi type 1 and Golgi type 2. The Golgi type 1 has longer axons, and the Golgi type 2 they have shorter axons. Depending upon origin, they are cranial and spinal nerves. Cranial nerves origin is from the brain. They are 12 pairs, and spinal nerves are from the spinal cord. They are 33 pairs. Based upon neurotransmitter secretion, they are adrenergic, cholinergic, GABAergic, dopaminergic neurons, and etc. Based on the diameter and the conduction of impulse, they are type A, B, C. This classification is called as Erlanger-Gasser classification. The type A fibers is further classified into alpha, beta, gamma and delta. This picture shows you the classification. Now we'll see neurotrophins. These are protein substances which help in growth and functioning of nervous tissue. Their function is initial growth and development of the nervous tissue and survival and repair of the nerve cells, and it also helps in neural transmission. The examples of neurotrophins are nerve growth factor, brain-derived nerve growth factor, ciliary neurotrophic factor, fibroblast growth factor, and etc. Now we'll see neuroglia. These are non-neural cells, non-excitable cells, and they are the supporting cells of the nerve system. They are divided into central and peripheral neuroglial cells. The central neuroglial cells include astrocytes. They help in blood barrier formation. They also help as tight junctions. It also helps in regulating the neurotransmitter levels. Now microglial cells. These are macrophages of the CNS. They help in phagocytosis of debris and infectious microorganisms. Next is oligodendrocytes. They help in myelination in the central nervous system, and it also provides support to the neurons. Now peripheral neuroglial cells. First is Schwann cells. It also helps in myelination in the peripheral nervous system. It plays an important role in nerve regeneration. Now satellite cells. It helps in supportive function of the neurons in the peripheral nervous system. Now we'll see properties of nerve fiber. They are excitation, conductivity, refractory period, summation, adaptation, infatigability, and all or none law. We'll see all these properties in neural communication. The neuronal communication is first accomplished via action potential, and the second is through the neurotransmitters, which is through chemically. And we'll see resting membrane potential, which is the static membrane potential in the quiescent cells. In nerve cells, it is of minus 70 millivolts. This potential is maintained by the sodium-potassium pump. When this resting membrane potential is disturbed, that is, when an external stimulus is applied, the sodium channels open and there's sodium influx, and also the potassium channels open and there's potassium efflux. Now the neuron is said to be depolarized condition. So at the end of depolarization, the potential would be from minus 70 millivolts to around plus 35 millivolts. The term Rio base means is the minimum voltage stimulus applied for a prolonged time will produce an action potential. Chronaxy means it is the minimum duration in which a stimulus applied is equal to 2 times the Rio base will produce the action potential, and utilization time is the minimum time that a current equal to Rio base will produce an action potential. The action potential generated at the axon hillock travels to the synaptic terminal. In unmyelinated nerve fiber, there is continuous conduction, whereas in myelinated nerve fiber, there is saltatory conduction of nerve impulse, whereas the nerve impulse jumps at the node of Ranvier. This saltatory conduction is faster in nature. There is a period in which there is no transmission of impulse. It is called as refractory period. It is of two types, absolute and relative refractory period. Absolute, there is no change even if the strength of the stimulus is maximum. In relative, there is a small change if there is maximum strength of stimulus is applied. Now summation. Summation is the addition of stimulus which produce action potential. There are two types. There is temporal and spatial summation. In temporal summation, when the presynaptic neuron is stimulating the postsynaptic neuron for multiple times, produces action potential. In spatial summation, when the multiple presynaptic neurons stimulate the postsynaptic neuron which produce action potential, is called spatial summation. Adaptation or accommodation, when there is continuous stimulus of a neuron, first the excitability increases, later the excitability decreases. This is because the sodium potassium pump gets deactivated on continuous stimulation. Infatigability, the nerve does not get fatigued even on continuous stimulus. All or none law. When one nerve is stimulated, it gives a maximum response or it does not give response at all. Now we'll see nerve degeneration and nerve regeneration. Causes of injury include when there's lack of blood flow, when there's nerve injury, when there's crush injury of nerve, local injection of toxic substances, There are 5 degrees of nerve injury. It was proposed by Sunderland. First grade is known as neuropraxia. Second degree is known as axonotmesis. Third degree is known as, and also fourth and fifth together they are known as neurotmesis. In neuropraxia, there's conduction block, and in axonotmesis, axonal injury takes place, and in neurotmesis, in type 3, there is endoneuron injury, and in type 4, there is perineuron injury, and in type 5, there's epineurium injury. Nerve degeneration, there are 3 types: Wallerian or anterograde degeneration, retrograde degeneration, and transneuronal degeneration. It could be anterograde or retrograde. First we'll see Wallerian degeneration, also known as anterograde degeneration, where the cut is present in the neuron. Distal to it, all the parts are lost, and in retrograde degeneration, proximal to the cut injury, everything is lost. In transneuronal degeneration, it could be anterograde or retrograde. Anterograde transneuronal degeneration, it is the degeneration of the distal neuron synapse to the cut neuron. Retrograde transneuronal degeneration is the proximal synapse neuron gets degenerated. Now we'll see nerve regeneration. This takes place only in peripheral nerves. The criteria for nerve regeneration is the nucleus and the soma should be intact, and the neurolemma should be present. The process of regeneration is the fibers distal to the cut injury undergoes Wallerian degeneration. Now the soma swells and the endoplasmic reticulum in the cell body degenerates, and the nucleus moves off the center. The axon stump now sprouts out into multiple processes. Now the regeneration tube is formed by the Schwann cells and the neurolemma present. Now the nucleus moves to the center. This is the anatomical regeneration, but physiological regeneration takes some time. And that's it. Thank you.